right, so I'm going to talk a little bit, kind of talk about basic stem cell culture and kind of some of the data we've generated in our lab in San Jose. I'm the product manager, but I kind of do a lot of R&D science still. My background is in assay development. I've done a couple of years of stem cell research, but I did a lot of NGS work in my previous job, and I know a lot of the. I did 2 years in my postdoc doing a lot of. We're looking at trinucleotide repeat expansion disorders, and we're working with stem cells from different DM1 patients, myotonic dystrophy 1 patients, and I understand the pain and the frustration, and going in every day to do your media changes and making sure that your cells are healthy. So I'm going to focus on a little of that, and I'll just start off by saying the promise really of stem cell research and iPSCs is one, that you can generate genetically and tissue relevant models. So being able to use these models for drug screening or for understanding mechanism of action or mechanisms of disease. And then the other is being able to generate these stem cells for potential stem cell therapies for degenerative diseases or cancer immunotherapies, and also metabolic diseases. And really, stem cells combined with advances in genetic engineering have really kind of made this field expand quite a lot. But there are a lot of challenges with stem cell line development, and I kind of summed it in two different buckets. One is that stem cell culture is very complex, and because of the complexity, you have more variability with your processes. So you have cell line variability, you have reagent variability, you have protocol variability, and then you have manual and user process error, which Dennis talked about earlier. But also, stem cells are more sensitive than kind of the traditional immortalized cells, or at least some primary cells. And so you have low cell survival and low cloning efficiency. I would also say that many of the tools that we use for stem cell research are kind of taken from standard traditional methods, and so those tools are really not as adequate for working with stem cells, and so there's a great need for more standardized processes, more defined and simple to use reagents, and more automated tools and processes to help streamline these workflows and improve the consistency. So I'm going to dive into two papers that were developed based on utilizing BioTechne's microfluidic dispensing technology. Some of you might be familiar with these papers, and they're developed at the NIH by Ilya Sinyak with Carlos Tristan and Yu Chen as the first authors of these papers. The first is really about trying to address stem cell sensitivity. So they worked on developing a cocktail they call the CEPT cocktail, which is a cocktail of small molecules that were used to help improve the outgrowth and improve the preservation of stem cells. And the second paper is really kind of an end-to-end protocol for how to generate cell lines using various technologies and techniques, and how to kind of have best practices in these approaches. So the first paper started with looking at about 15,000 compounds and doing kind of a basic high throughput screen where the cells were plated out, the compounds were plated out in 1536 well plates, and HES cells were plated on top, and basically they looked to see, are there compounds that helped these cells grow out and not die in the plate, basically. And they used the cell titer glow assay to do this. No problem. And they simplifying greatly their process, but they did some rounds of combinatorial screening to identify 3 compounds, chromin-1, imricasan, and trans-ISRIB. And chromin-1 was found to be a very, very potent ROCK inhibitor, much like the traditional Y27632 compound, but actually has the IC50 in the picomolar range, whereas the Y27632 compound is in the nanomolar range. So a much more potent ROCK1 and 2 inhibitor. Imricasan was found as a pan-caspase inhibitor, so inhibiting apoptotic processes. And the trans-ISRIB compound was identified as an inhibitor of the integrated stress response pathway. And there was some empirical evidence that they found that polyamines or these polycations were known to support cell growth and stabilize structural integrity of the cells. So they generated some experiments with different combinations and comparing these to kind of the traditional approach of using Y27632. In this first experiment on the far left, they did an outgrowth experiment where they did a minimal plating of 25 cells in a well, and they compared the CEPT cocktail with variations of CEP or CET or just CE, and also compared it to Y27, and found that the CEPT cocktail had a better outgrowth efficiency with this minimally plated HES cells. In the figure to the right of that, they showed that the CEPT cocktail showed the cells had a greater attachment affinity to the plate. So this is an impedance-based assay where basically the plate has electrodes on the well, and when the cells attach to the bottom of the plate, it increases the impedance of the plate bottom. And as they form tight junctions, that impedance goes even higher. So they show that the CEPT cocktail gave the highest impedance of these cells attaching to the base of the plate, or to the matrix. And then finally, important for us is that they show CEPT compared to the Y27632 compound with various cell lines, where they did a single cell deposition using BioTechne's an earlier version of the PALA called the HANA. They did a single cell deposition with these cell lines with either the Y27632 compound or the CEPT cocktail, and found the CEPT cocktail was more efficient in growing out those single cells into clones. So this is, this cocktail showed that, there's really was trying to address kind of developing tools to help improve the sensitivity aspect of single cell outgrowth with stem cells. In the second paper, they really kind of developed this end-to-end workflow, going from thawing all the way through clone characterization. And it's a great paper. We utilize this in our lab to kind of develop our protocols and process, starting with thawing and passaging and maintenance, and then going into either, if you're editing cells, or if it doesn't have processes around editing, it kind of moves into just single cell dispensing. But if you're editing cells, then you would go into the single cell dispensing, and then confirming single cells in the plate using microscopy, following that clonal outgrowth and maintenance, and then expansion and clone characterization. So I'm going to kind of go through each of these steps in a little more detail and talk about some of the best practices that they introduced and talk about in their paper. So for some of you it might be basic, and you might say, of course, that's obvious, but we're here all to learn, and everyone's at different levels. So please ask questions or speak up if you have any questions while we go through this. I'll just mention that they did use in the paper, they used various kinds of stem cell reagents and products. When we do this workflow, we're BioTechne, we want to use our products and develop our products, and so we used our own Accelerate iPSC expansion media and our own Cultrex ready basement membrane extract. The great thing about our media is that they're animal and human component free, so they're really developed for GMP workflows and for translation. They're an all-in-one format, so they're very simple and easy to use, and they support every process through a stem cell or iPSC workflow, thawing, passaging, and single cell cloning. And then our Cultrex Ready BME is a basement membrane extract. It's pre-diluted and ready to use right out of the fridge, so you don't have to, you get those little vials of matrix gel, you don't have to then dilute them and add more variability to your workflow. These are just ready to use right out of the fridge for coating plates. So as Katai mentioned, one of the important things in iPSC workflows is minimizing stress on the cells. And thawing is a very stressful process for cells, specifically stem cells. So it's not just as easy as taking them out of the freezer, throwing them into the water bath, throwing them into your media and plating them. We want to minimize mechanical stress, so we use wide bore pipettes. We don't vortex or pipette up and down with our cells. We really want to avoid breaking the cells up from that aggregate state. We want to keep them as kind of little rafts, little aggregates. And then when we're performing our first media addition, we really want to minimize osmotic stress. So we perform this by adding the cells to a small or a 15 mil conical tube first, and then slowly adding your media one drop at a time to gently kind of bring them back into that media while you're kind of gently agitating the tube. We want to do this with either a ROCK inhibitor, traditionally it's the Y27 compound, but in this paper and this workflow, utilizing the sept cocktail has been shown to be superior, and so this inhibitor is used to minimize apoptosis during thawing, and it's used in the media only for the first 24 hours of culture, so it's not used after that unless you're passaging, which we'll talk about. Another important part about this is really you don't want the cells, when you're thawing them, you don't want to thaw them at too dense in the plate or too sparse, because that can increase differentiation of your stem cells. And so you do have to, when you're working with new cell lines, you do have to kind of optimize how densely you thaw your cells onto your coated plate. And so it's recommended that you start with titrating that to see what works best in your lab. So with passaging and maintenance, so once you've thawed your cells, your first media change after 24 hours would include no ROCK inhibitor or CEPT cocktail. And as long as your cells are you're not removing the cells off the plate, you wouldn't need apoptosis inhibitors just when you're doing daily media feeds. But once your culture gets to 60 to 80% confluence, that's when you want to basically do your first split. And splitting is also kind of a technique that you need to optimize as well. So we've optimized our split ratios. We tested 1:5, 1:10, 1:15, and 1:20. We found in our lab 1:10 worked best, and it's important because if you split at too hard, or if you split you allow the cells to be too dense, that can lead to more cellular differentiation in your culture. And whenever you're passaging cells, we really don't want to break the cells up as single cells during this process. We really want to keep that mechanical stress on the cells very low, so we avoid breaking those aggregates up, and we use just a simple 0.5 millimolar EDTA solution, which is a metal chelator, so it helps the cells to break loose of the attachment and basically creates kind of small clumps of cells. We passage those clumps rather than breaking them into single cells in this phase to help them be less stressed out. Whenever we do a passage, we also utilize CEPT during this first 24 hours, but then after that, again, it's removed after the first 24 hours during that first media exchange. Tristan et al. In this paper, with developing this process, they recommended before doing any cloning with your cells to adapt your iPS cell culture for at least 7 passages with this cocktail to help improve the cell quality. And so once you've kind of established your culture and you want to move into dissociation and single cell cloning, we always recommend staining with viability stains. This helps you to both dispense viable cells. So the viability stain of our choice is calcine AM. We've tried some of the branded like Cell Tracker, for example. It's a thermo product, not bad-mouthing it. We use it for all of our other cells, but they didn't work as great. It didn't work as great for iPS cells. We got a much higher signal with calcine. And we can do this directly into our BioTechne Accelerate media because it doesn't have it's a defined media, so it's lacking a lot of the esterases that would cleave up your stain before they would go into the cell. So calcine is an inclusion stain, so that means that it's staining live cells and gives you the ability to also, when you dispense the cells, to be able to visualize them in the plate and help you to determine if you have a single cell in the plate or multiple cells in the plate. For dissociation, so moving from EDTA, we use Accutase for single cell dissociation. So this breaks the cells up into single cells. And we also filter through a cell strainer to remove out any larger aggregates. As I mentioned, we dispense using calcine to gate for live cells. But the great thing about the Paula is because you have multiple fluorescent channels, if you wanted to select for a specific reporter construct that you're expressing, say mCherry or Pacific Blue or something like that, you could stain for that and increase the selection of your cell line that you're trying to develop. So we have many customers that might use 2 or 3 different stains to select for the population that they're interested in. And once you're ready to dispense, you'll see this in the lab, but we dispense into matrix-coated plates filled with growth media. I'll talk about in the next step. You don't want to put too much growth media in your well because in the next steps you're going to feed your cells, and you want to really minimize you're not going to remove, you're actually just going to add on top. So it's important to get the right amount of media to allow that first, second, and third day of feeding. So with Paula, we've done some deposition experiments with iPSCs in the last couple of months in our lab in San Jose. And we used 2 different vendor cell lines that we were working with. We dispensed multiple plates with multiple cartridges, and we compared that with just doing hand limiting dilution. We found that on average we see about 3-fold more single cells per plate than just limiting dilution. And there's some variability that we attributed actually to the 2 different cell lines. So with dispensing, we found that the vendor 1 had a slightly lower number of single cells dispensed than the vendor 2 cell line. So these cell lines can be very different in terms of their sensitivities, and so you have to kind of optimize with your particular cell line. You can get very different kinds of results depending on the cell line you're working with. So, in order to quantify single cell clonality, you really need to use whole well time course imaging. So once we've dispensed the cells, we allow the cells to settle for about 2 hours, and we image day 0 through day 10 to characterize the clonality and also the growth rate. We use both fluorescence and bright field imaging. And these two microscopes are the ones that we use in our lab, so the CountStar Castor X1 and the Synintech Ni1. We utilize both of them. They're both great products. We don't make them, but you can see here just one image of a whole well with a 4x image. It's hard to see it, but here up in the corner is your calcine stained cell. One of the tricky parts that you'll find with imaging cells is when they do get into the edge of the well, it can be very difficult to determine clonality. And so that, with having fluorescent stains, it does help to be able to determine that. But if you were to look for that cell just under bright field, it becomes very impossible. So how these products work is basically they track, so as you image over time course, and as the colony grows out, you would then kind of backtrack to day 0 where that colony was to look for your single cell and determine the clonality of that cell line. And that's how both of these products, both of their algorithms work. So we found colony outgrowth and maintenance. As I mentioned, on your recovery time, after you've dispensed your single cells, we don't take any media out of the plate. We actually only add for the first 3 days. So we add 50 to 100 microliters of growth media without CEPT through day 1 through 3. And we want to, again, avoid mechanical stress, avoid perturbing the cell as it's attaching to the plate. So you need to make sure you have some headspace in your well on the first 3 days. And then once you go into the outgrowth phase, days 4 through 10, you want to basically do a half media exchange where you're removing half the media and replacing it with an equal volume of media, but again without CEPT as you're doing as you're in the recovery and outgrowth phase. When you're doing these media exchanges, it really helps to have some automated liquid handling technology, as you don't want the well tips or the tips to go down to the bottom of the well and perturb your culture. So really it's very important to have some kind of automated pipette tool here, which Dennis will be able to show you what they use here in the lab. Just mentioning the iPSC clone outgrowth, so this is from the data of the dispensing data that I showed previously. So again, we see on average, threefold more clones per plate than using a standard limiting dilution approach. And so each dot here represents a 96 well plate. So threefold gives you, basically, with these particular cell lines, gives you the ability to improve the efficiency of your workflow. I will mention that there's quite a lot of variability that we've seen comparing our data to Dennis's data, for example. Dennis can get anywhere from 20 to 50% outgrowth with his iPSCs with using the Paula. These are just two different cell lines that we were using, so we were seeing about, on average, about 15 to 25% or 30% outgrowth. We actually looked at comparing ROCK inhibition versus between the CEPT cocktail and the Y27632. And we actually found that one of the cell lines, it didn't matter which you used. If you used Y27632 or CEPT, it seemed to be give same outgrowth rate on average. Whereas the other cell line actually did not grow out at all, almost, with the Y27632 compound, but did have a significant number of clones with the CEPT cocktail. So again, showing you some of the variability that you might experience with your iPSC cultures, and having the CEPT cocktail gives you that other tool to help you to improve the outgrowth of your cell lines. Finally, once you get to the clone characterization and expansion phase, we recommend, or the paper recommends using pluripotency staining. So first you can use TRA160 staining. We again had to titrate and optimize the incubation time of this antibody. Looking at cell morphology and cell shape is also another way to characterize the health of your clones. And then they recommend expanding your TRA160 positive clones and passaging these into a matrix-coated 24-well plate, and using the similar processes that I described previously, and to also, at this point in time, start to cryopreserve your cells once they've reached 60 to 80% confluence, prior to any further characterization that you do. This was an experiment we did comparing the health of the PALA to limited dilution acquired clones. So if you imagine, limiting dilution is probably the gentlest method that you could use for dispensing a cell into a plate, and the PALA is this automated tool. So we wanted to look, is the PALA inducing any additional cell stress or strain on the cells? And we show that the clones that were acquired with PALA versus limiting dilution grow the same, have the same outgrowth. So in terms of clone area, as we measure the clone size, and then also looking at TRA160 expression, we found that the clones derived with PALA had a very similar profile of the TRA160 marker as the limiting dilution acquired clones that we had as well. Finally, when you're running analysis of your clonal cell lines, we want to do karyotype analysis, looking for any aberrations in chromosome numbers, looking at whole genome sequencing, looking at your mutation rates. Alkaline phosphatase staining is recommended. And then, of course, looking at pluripotency markers such as OCT34, NANOG, and SOX2, and then doing germ layer differentiation into ectoderm, mesoderm, and endoderm. BioTechne offers multiple kits and products to help support this workflow, including our functional identification kit, as well as our immunocytochemistry kit that has the reagents you would need to do the staining for the pluripotency markers and the germline differentiation. So again, just throwing this out there, we have these products that are used to support your workflow. And in summary, stem cells hold enormous promise for developing disease-relevant cell models, and developing more stable, more consistent products. They're here and they're quickly improving all the time. And BioTechne offers a range of these products to support your workflow. And with that, I'll turn it to you for some questions.